Okay. Chemical control? I sweat and shake. Hear me moan! Oh no! My hormones! Yeah. So the history of the endocrine system could take several lectures itself, of course. Some pretty interesting stories that are in here. I'm just going to give you one slide of them. I will talk a little bit about a guy named Seal at the end if I get there. Probably won't. He's a pretty interesting guy too. But Chinese medicine has been familiar with aspects of the endocrine system for literally thousands of years. Again, we often think of medicine as being highly Eurocentric or even just the Mediterranean, but talked about Sushruta from India. The Chinese pharmacopoeia was clearly the most sophisticated at that time in history. And in this document here, that's part of the Chinese pharmacopoeia called the Inner Canon of Huangdi. I'm not sure if I'm saying that correctly. I think I am. Huangdi. They describe various methods for isolating prostaglandins, pituitary hormones from various sources, often from urine, and could use them for the treatment of different types of ailments, such as prostaglandins were used to treat sore throats. So there's this is just the tip of the iceberg, quite an extensive understanding of various endocrine components in ancient medical practices in China. And let's fast forward 2,000 years to where the Europeans began to catch up. This guy, Arnold Berthold, noted that cockerels, these things down here are called cockerels, they're roosters that have been castrated. And you do that, you castrate a cockerel because it makes them less aggressive, first of all, and it also makes them put on more weight. They put on much more weight. So rather than kill the bird, if you want to use it for meat, you can castrate it, and it will become a more succulent bird. He noticed that when these roosters were castrated and became cockerels, that their combs on the top and their wattles below them, sex-linked traits on these birds, did not develop. And they also, as I had mentioned, were not as overly aggressive. And this guy was at Göttingen University, and he was the first person to attribute that to some sort of chemical component that was being removed, some chemical factor that was being removed when the testes of these birds were being removed. And that the effect of that chemical component, that hormone, is what led to these sex-linked traits. So it wasn't just something genetically intrinsic, that there was a chemical factor that was being produced. That was sending a signal to the rest of the bird that led to the development of their male traits. That's really the first modern recognition of chemical signaling. All right, we'll leave the history there. And just first of all, let's get a grip on the anatomy. Well, as I've said, it's a bit of a pyramid with the hypothalamus and the pituitary at the top of this pyramid. So the hypothalamus produces ADH, which we've talked about, oxytocin, which I only mentioned, I believe, in the context of one of my first lectures when I was talking about childbirth and contractions and the effect of oxytocin on the uterus, but also produced by the hypothalamus. And like a host of what are called regulatory hormones, actually releasing hormones is what they're called, and we'll see the effects of those in a bit. And then there's the pituitary gland. We'll look at the anatomy of the pituitary a little bit more, slide or two. But it's broken up into two. Actually, really, there's three parts to it. There's the adenohypophysis, which is the anterior lobe, the neurohypophysis, the posterior lobe, and there's a thin slice in between them called the pars intermedia, which we won't talk about really, but that's no matter, just so that you know that I'm not deceiving you. Anterior lobe is it's kind of the chemical factory of the pituitary. It produces adrenocorticotropic hormone, thyroid stimulating hormone, growth hormone, prolactone, follicle stimulating hormone, luteinizing hormone, etc. And then the neurohypophysis, this is the one that you saw in the diagram in the kidney that had the portal system, the blood flow going through it. And this is where the oxytocin and antidiuretic hormone that are produced in the hypothalamus are released into the bloodstream. And then everything else, these other target hormones, target endocrine tissues in the body are widely distributed. There's endocrine tissue all over the place. So the pineal gland is still in the brain. The pineal gland is responsible for taking tryptophan, making serotonin from that, and serotonin gets converted into melatonin in the pineal gland. Pineal gland is indeed sort of your third eye, if you want to think of it that way. And does anyone into lizards, like what are those called? Iguanas? Everyone been to like an island in the Caribbean somewhere that's overrun by iguanas? Yeah, well, if you ever held an iguana and looked at it on the top of its head, right, kind of where your apple core would be up there, iguanas have something called the pineal eye, and essentially iguanas don't have this huge cauliflower cerebral cortex on top of their brain the way we do that has buried our pineal gland. Their pineal eye is actually exposed to the outside world. And there are light receptors in both theirs and ours. They're non-image forming. We don't sensate through them. They are photoreceptive and regulate circadian rhythms. So melatonin is a hormone responsible for mediating circadian rhythm. And you can, like, for example, has anyone ever taken melatonin before? You can buy it over the counter. It's sold as a sleep aid. It's probably not good to take large quantities of melatonin, and also not good to take it for long term because it can throw off the endogenous melatonin production in the pineal gland. But it can help if you have jet lag, get your system back on. And train it to the new the circadian timing. Other ones, moving down the axis, we have the thyroid and parathyroid glands. We've talked about parathyroid quite a bit in the past when we were talking about calcium homeostasis. The thyroid gland, we may or may not get to talk about it on Wednesday, but this is where thyroxine and T3, T4 are produced. We have talked about calcitonin, which is produced by the thyroid. The thymus, easily confused with the thyroid gland. They kind of look the same. They're in a similar part of the body. They're both kind of cradling the trachea there, but they're very different. The thymus is primarily active during early infancy, just postpartum during thymic deletion. I think I've talked about that in here. Have I talked about the immune system and thymic deletion just a tiny bit. Maybe I didn't. Well, anyways, it's responsible for training your immune system and killing off T cells and B cells that produce antibodies that recognize the self. It produces hormones called thymocyns. And atrophies as you move into childhood and then adulthood. The heart makes the natriuretic peptides, ANP, which we talked about a little bit, and BNP. Kidneys, we covered pretty extensively. Suprarenal glands. So we'll talk about those a little bit if I can get to the end of this lecture. But we have the medulla, which makes epinephrine and norepinephrine, and the cortex, which makes the cortisone derivatives, corticosterone, cortisol, the related aldosterone, and then the androgens. What else? The adipose tissue, which is all over your body, makes leptin and ghrelin, which we may talk about a little bit in the GI chapter. The GI tract also has numerous hormones, which we'll talk about when we get there. The pancreas. We will talk about the pancreas, which is kind of the main thrust of my endocrine chapter here to get us ready for the GI tract. We'll talk about the pancreatic islets and the production of insulin and glucagon, which will also help you for your IR AMPK talks. And then there are the gonads, the testes and the ovaries, which we will sadly not really get to cover at all. But no pun intended. Any questions on the anatomy? You got it. Okay. So just to remind you of the ways in which we are able. So I told you that homeostasis, in the beginning of the year, I told you that homeostasis is intimately dependent upon whatever form of communication is being utilized, right? So I said there was electrical via the nervous system, and then there was endocrine, right? And so let's review that a little bit. So there's direct communication between adjacent cells that are directly what is called synstitial. They're directly attached to one another via gap junctions. Remember gap junctions? I talked about those. The little like grommets that hold two cells together that allow small solutes to pass between adjacent cells. So this can be small solutes, various lipid-soluble materials that may be in tiny vesicles, ions, etc. So it has to be cells that are directly adjacent and attached via these gap junctions. Next is paracrine communication. So paracrine communication is to nearby cells that are different from one another, and they signal through the extracellular medium. So whatever paracrine cell we're talking about releases its hormone, its whatever chemical factor, and the other cells that are then sitting in this broth absorb those chemical factors and lead to whatever response that we're trying to elicit. I kind of cut out, there's no autocrine on here, but autocrine was another one of these very local mechanisms. It's like paracrine, except its release and effect onto the same type of cell through the extracellular fluid. And then there were the two methods of systemic, system-wide control. And they were chemical, which was the endocrine, and neural or synaptic communication. So we covered the nervous system, that's on the bottom. And then the endocrine communication involves the release of hormones from synaptic vesicles, intracellular synaptic vesicles, directly into the circulatory system, where they'll circulate around the body and activate whatever target organ, whatever effector is the target for the hormone that has been released by the endocrine tissue. Okay, basic, simple review. Okay, so here I probably won't get a chance to completely walk through this today, but this is my attempt to organize hormones for you. There's the structural organization, and then there's a functional or a mechanistic organization of them on the other side. So hormones can be broadly categorized into three groups. There are ones that are amino acid derivatives, highlighted in blue towards the top, and they consist of really only two types. Ones that are made out of tyrosine, and those are the thyroid hormones, or produced by the thyroid, T3, T4, or the catecholamines. And the catecholamines are produced either by the suprarenal medulla, or dopamine is made in the brain. And then the other amino acid derivative is tryptophan, melatonin, that we had talked about a little bit. The second category are, rather than single amino acids, they're peptide hormones. And you can be roughly broken into two categories based upon the chain length of the peptide chain, right? So it can either be short polypeptides under 200 amino acids, and this lower purple category, or if they're larger than 200, you're going to call them a glycoprotein. And then lastly are lipid derivatives. So these are ones that are made out of fats, of course, and they break into two categories. They are eicosanoids, and there's several of those. I mentioned prostaglandin, the prostaglandins earlier when I was talking about the history of Chinese medicine, and they're a lipid derived hormone. So all the eicosanoids are originally made out of arachidonic acid, which is a type of fat. And then there are the steroid hormones, which are made out of cholesterol. And there's several of those, including various androgens and estrogen. All right. So mechanistically, there are two modes of communication that hormones can use. They can either start a signaling cascade where the hormone binds extracellularly, and that binding event on the surface of the cell is somehow translated to the interior of the cell by a mechanism called G protein coupled receptors. I'm sure many, if not most, who has talked about G proteins before? Yeah, a couple of you. Maybe half the class. Good. Well, G proteins can have an effect either on cyclic AMP levels or on calcium, and we'll break that down and look at that in a bit. The second broad category is intracellular. So there are hormones that in these structural categories that are able to pass directly through the membrane. And as you may intuit, they are going to be lipid soluble. So this is going to include any of the lipid derived hormones, the steroid hormones, and also the thyroid hormones are quite lipophilic and able to pass directly into the cell and affect their signaling intracellularly. So we're going to walk through all that one step at a time. All right. First, hormones that work extracellularly to affect cyclic AMP levels in the cell. That includes all of the peptide hormones. If it's a peptide hormone, it's going to affect cyclic AMP. Melatonin also affects cyclic AMP, and any of the catecholamines also. All right, so how does this work? Hormone binds the G protein coupled receptor. There's some sort of protein receptor that is coupled to a G protein. This G protein is activated when the hormone binds, and that G protein can activate adenylate cyclase, which is a hormone that takes ATP. It takes one mole of one unit of ATP and hydrolyzes 2 phosphates off of it and then cyclizes it. I don't have a chemical structure of cyclic AMP on here, but it's no matter. Anyways, cyclic AMP itself is what's called a second messenger. The first messenger was the original hormone. Maybe that's melatonin, or maybe it's dopamine, or whatever it is. The first messenger is the hormone that binds to the protein receptor on the outside of the cell. And then there's this transduction of that signal to the inside of the cell, where ATP is converted into cyclic AMP. And cyclic AMP, I have just a few arrows here saying activates kinases, and those kinases open ion channels and activate enzymes. This is again like the great simplification of what can be super complex pathways, right? Cyclic AMP signaling, every cell, every signaling cascade that's being touched by cyclic AMP is going to have some large cascade to it. But cyclic AMP gets activated, and that acts as the second messenger in whatever cellular context it finds itself. So there can also be the activation of phosphodiesterase. If a hormone, so the hormone can either turn things on or it can turn things off. Right? So you can have a different hormone bind a G protein coupled receptor that is instead coupled to phosphodiesterase. Phosphodiesterase is going to cleave cyclic AMP to simply AMP. And this is going to sort of turn the system off. So there's one hormone, G protein coupled receptor, that can upregulate cyclic AMP. There are different sorts of pathways that can turn cyclic AMP down. You turn it on via G protein activation of adenylate cyclase, and you turn it off by G protein activation of phosphodiesterase. Adenylate cyclase and phosphodiesterase are kind of acting in opposite regard in the cell. Does that make sense to everybody? All right. So you can also have a similar sort of motif using calcium as the second messenger. And the way this works, rather than using adenylate cyclase or phosphodiesterase, which is bound to the G protein, it can activate phospholipase C, which is a protein that will stay associated with the membrane. And phospholipase C goes through a little pathway that leads to diacylglycerol that can then activate protein kinase C. And when protein kinase C gets turned on, it will open up these calcium channels calcium floods in, and calcium acts as the second messenger, combined to calmodulin, and calmodulin can have a bunch of downstream effects in the cell. So, phospholipase C also is able, through another branch in the pathway there, to activate IP3, inositol triphosphate, which is also going to release intracellular stores of calcium. So calcium can either come into the cell through the channels via protein kinase C, or be released from intracellular stores in vesicles, endoplasmic reticulum, via the action of inositol triphosphate. So that's the broad outline of calcium signaling. Now you'll see when you dig into the insulin receptor diagram, you're going to see this motif. You'll see this motif there. So what are some examples? Oxytocin works this way, epinephrine and norepinephrine, the other hormones of the hypothalamus, and many of the fish oil-derived hormones, the eicosanoids. All right. You guys all still with me? I forget my phone. Then there are the intracellular hormones, the steroid hormones being the first. So here is I don't know what hormone that is. It's just a general schematic. It kind of looks like cholesterol because the steroid hormones are made out of cholesterol. It's able to pass through the lipid membrane, and it's able in the cytoplasm to bind some kind of a receptor, some sort of receptor. And that can either be in the cytoplasm or it can be directly in the nucleus, depending on the cell. But this hormone binds this receptor, which is often a transcription factor, which then enables, so the steroid hormone's like the key that turns this transcription factor on. It's able to bind to a strand of DNA and then turns that DNA on for transcription into mRNA, and the mRNA is then exported into the cytoplasm for protein synthesis. All right. So, and then whatever the downstream effects of that are. Some examples of this are estrogen, progestin, any of these steroid hormones that look like cholesterol. So I think that clock is still a minute fast. I think I have 1 minute. I don't have my watch on me. Anyways, the last of them, I'm just going to do it. This is the last of this sequence here, are the thyroid hormones. So the thyroid hormones themselves are able to translocate across the membrane in much the same way as a steroid hormone. Again, binding to receptors either at the mitochondria or in the nucleus. So thyroid hormones can have an effect. Are you all aware that you have, there's the DNA that's in your the nucleus of your cells, but then there's a whole other set of DNA that you have in your mitochondria? Is everyone aware of that? Yeah, and you only get your mitochondrial DNA from your mother. Your nuclear DNA is from your mom and your dad, but your mitochondrial DNA is exclusively from your mother. It's these thyroid hormones that affect the DNA in your mitochondria and ATP production, etc. Or thyroid hormones can go to the nucleus and act in much the same way as the steroid hormones by binding a transcription factor, turning on those genes with a similar cascade of protein production. All right. Are there any questions on any of this? That's class.